Moving on. Okay, yeah, it was her dinner. Anyway, okay. Gram-positive, stain purple, because they have all that outer layer of peptidoglycan, and they're of course susceptible to penicillin. So, all right, that's where we left off. Um, what about that penicillin remark? Why is that? I mean, okay, so what is penicillin doing to the bacterial cells? I'm so glad you asked. Okay, so penicillin, like I said, targets a little enzyme that links together the peptidoglycan cell wall. Okay, so what is this enzyme called that links together the peptidoglycan cell wall in bacterial cells, Miss Eden? So glad you asked that too. So the name of the enzyme is the PBP enzyme, and it is a big antibiotic target. Okay, big in that it's a favorite of pharmacists, okay? So the PBP enzyme, let's talk about that. So let's pretend PBP stands for peptidoglycan building protein. And again, it's an enzyme because, again, what does everything for a cell? An enzyme. So there, other enzymes that are involved in bacterial cell wall production. Sure, there's a lot of steps, but we did know how to target the PBP enzyme with penicillin. So over here is a schematic, and this is supposed to represent the sugar bar. So nag, nam, of course, the glycan part of peptidoglycan. And what are the peptide parts? Well, those are the peptide chains here. And you see there are 5 little amino acids. So where's the PBP enzyme? Glad you asked. So this is the PBP enzyme in gray. So what it does, so you can see it has an active site here. Remember Mr. Seberg's lecture? I know the enzyme looks different, but here's this group. So what is the substrate for the PBP enzyme? Well, that would be some of the amino acids. So the amino acid chain is the normal substrate. And then what happens is the enzyme links together one chain with another. And again, it helps to link together peptidoglycan, sorry. And again, PBP enzymes are only found in bacteria. The function again, it links cell wall peptide chains together. Okay, and again, the peptide chains are the enzyme substrates. So without PBP linking together the peptide chains, the cell wall is weak and the cell can undergo cell lysis. Water rushes in through osmotic pressure and it'll explode without a cell wall there. Because this is a bacterial cell wall, well, this probably isn't going to break our hearts, right? So it makes sense to target the PBPs and shut them down. All right, so a little more about PBP. Does PBP really stand for peptidoglycan building protein? No, it really stands for penicillin binding protein, even though its job is to build the peptidoglycan. Okay, I always start this way with my students because it's confusing. Why did they name it penicillin binding protein? Because they never knew about this enzyme. They were just trying to figure out what's going on in the bacterial cell that gets blocked by penicillin. And they said, oh, so penicillin's killing these bacteria because it is a competitive inhibitor of this little enzyme. So we'll call the enzyme the penicillin binding protein. But that doesn't tell you what this enzyme normally does on a good day for the bacteria, okay? It builds peptidoglycan. So penicillin interferes with the peptidoglycan production. So we'd say penicillin is a competitive inhibitor of the PBP enzyme, right? So here we have, let's go through the chain here. So again, here's what the normal PBP enzyme does. It's supposed to link the chains together, but bring in penicillin. Now, it normally doesn't look like a hockey puck, but here it is in kind of orange. And part of the penicillin molecule fits into the active site, right? And it's going to outcompete the normal substrate. So it's a competitive inhibitor. That's what penicillin does. So the penicillin is a competitive inhibitor of the peptide chain substrates that normally fit into the PBP enzyme active site. So all that stuff you were learning with Mr. Seberg and you thought you'd never have to come back to it. Aha! We're back. So you need to know. So again, here is, now stop for a moment, picture a bacterial cell. In the outer edges of the bacterial cell, in the envelope, you have the peptidoglycan cell wall, which is like scaffolding. Okay, how is it going to be manufactured? You need an enzyme. What's the name of the enzyme? PBP enzyme. So its normal substrate are going to be just the peptide links, the peptide chain, and it just links them together. That's what the enzyme does. Now, penicillin gums it up. It jams itself into the active site. So it's a competitive inhibitor. Okay, if you have trouble following this, give me a call or send me an email, okay? So again, penicillin stops the linking of protein cross bridges. So what happens to the cell wall? Well, okay, if it's a gram-positive bacterial cell, it's really going to be in deep trouble. If it's gram-negative, it may work, believe it or not. Even though the outer membrane does inhibit a lot of things from getting into a gram-negative bacterial cell, some with like Neisseria gonorrhea and Neisseria meningitidis that causes meningitis, you can use, well, at least in the old days, you used to be able to use penicillin, so it just depends. But again, the outer membrane, usually of gram-negatives, block the access of penicillin, okay? So here we have an animation, a video, actually, a movie. And these are bacteria, and they are dividing by something called binary fission. So, of course, these are bacilli. They're rod-shaped. Now, as the bacterial cells grow, they have to make more cell wall, don't they? They're growing. They're building stuff. So there's going to be lots of PBP enzymes out there linking together the sugar bars. Okay? And even if water rushes into the cell, they're fine. They've got a strong cell wall. So the pressure can press the membrane up against the cell wall, but the cell will not explode. What if we add penicillin and it'll enter and it will block the poor little PBP enzymes that are linking together the cell wall? What could happen? Okay, look to the left. Okay, let's watch. Okay, yeah, there's not much. Okay, there it is, recycling. And I'll have to do sound effects. And, right? So, all right, my sound effects aren't really good because I had a root canal this week. So, but you can see where penicillin is quite the killer. It will kill bacteria. Not all antibiotics kill the bacteria. Some antibiotics just slow them down so the immune system can take over. So without penicillin, with penicillin. And this is what happens when the cell wall can't link together the peptidoglycan. Okay? The little PBP enzymes aren't doing their job. All right. Now let's switch a little bit. We're still going to talk about cell envelopes, but somebody asked the question, hey, I heard that the TB bacterium is acid fast. What is that? I'm sure you all asked that question, right? Well, okay. It's still about the bacterial cell wall and the envelope, so we'll continue with that. Oh, that's not good. Okay, but we're talking about the cell wall and cell envelope here. So there's another differential stain. Now this stain has different chemicals with different colors, totally different. So erase the gram-positive, gram-negative thing for just a moment. I will slightly refer to it, but just different colors, okay? So the acid-fast stain, again, is different than the gram stain. It's a different test, different color, different chemicals, and different colors. Now, it turns out that some bacteria have a very unique and very protective waxy coat, and that waxy coat is made of a substance called mycolic acid, and it surrounds the cell wall, okay? So looking down here, here's some bacterial cells. These are really weird bacilli. They're all pink, and the rest of the material is not pink. But if we looked at the makeup of the cell and cell envelope. This is the inside of the cell, the cytoplasm. You have the normal plasma membrane. You have lots of layers of peptidoglycan. There's no outer membrane. So this is gram-positive, but what it has is a very protective layer of mycolic acid and some other lipids. Okay. This makes this organism very, very, very tough. Hard to kill. What penetrates this stuff? Not a lot. Okay, now the bacteria that have this mycolic acid are actually named after the mycolic acid that's in their envelope, just outside their cell wall. So Mycobacterium leprae causes leprosy. Mycobacterium tuberculosis. Yeah, you got it. Causes tuberculosis. So think about how protective that is. And let's say if someone has active TB in your, on your floor, you better be really good with your universal precautions. And yeah, it's going to be tough. Okay. So to identify the mycolic acid, we use a special little stain and staining process. So we call the process, the staining process, the acid-fast staining procedure. The first stain is the one that is going to attach to mycolic acid, and it just happens to have a red color, but it's carbylfusion. So red carbylfusion, I know, gesundheit. And this attaches to mycolic acid and nothing else. Now, when you decolorize, everything else goes clear, and then we want to see those other things. We don't want them to disappear, so we can counterstain with methylene blue for anything that isn't acid-fast. So here we are. And here's the red carbylfusion. You can see it stained the bacterial mycolic acid and nothing else. So you look at this in a sputum sample and you go, uh-oh, right? Patient probably has tuberculosis. So in this case, anything red, carbylfusion red is acid fast. The blue is non-acid fast. Okay? So how do you kill an acid-fast bacterium that is so protected? By the way, over here is a lung that was infected with Mycobacterium tuberculosis, and it's in bad shape. Well, you better catch it early, and you better treat it the right way. One way is to use an antibiotic that blocks an enzyme that makes mycolic acid. You know, like before we blocked the enzyme that made peptidoglycan. Okay, this one we have to stop mycolic acid. This is really, really busy. You don't need isoniazid. It's not in the terminology. But this is one of the drugs as well as some others that you can take. So down here, there's ethambutol and some isoniazid. But okay, so if this is the Mycobacterium tuberculosis, and here it is again, here's the normal membrane, the peptidoglycan, and here's the mycolic acid. So it stops the manufacturing of mycolic acid by blocking the enzyme called catalase peroxidase enzyme. I know. But again, this is one way. But what you do need to know is if you wanted to block this particular organism, you can stop it from making mycolic acid. But I'll tell you what, these bacteria mutate so very quickly that you have to use other drugs that do other things. So you need to sometimes combine it with a drug that knocks out the enzyme RNA polymerase. If you remember that, that's important in DNA transcription, right? If you're going to make proteins, maybe you want to add that with a few other drugs. So usually you start off with 3 drugs because these little guys mutate and it's hard. You need to get the drug to them and you need to get several kinds. Okay. Back to the just regular cell wall stuff. Some bacteria do lack a cell wall. How do they get away with that? Well, okay, they don't break apart because they have extra special stuff in their plasma membrane, not cell wall, plasma membrane. The bacteria that lack a cell wall, I mean, there's several kinds, but the one I'm going to mention is known as a mycoplasma. Mycoplasmas include Mycoplasma pneumoniae. Does it cause pneumonia? Yeah. It causes something called walking pneumonia. So because it has no cell wall, no peptidoglycan, no PBPs, penicillin, does it affect these bacteria? No. But if you're thinking, well, what keeps them from exploding? They've got something again in their plasma membrane known as, okay, they have extra sterols in their membrane, and that gives it strength so they don't explode. By the way, these things are really weird. So here's a little circle. There's one bacterial cell. If you had to characterize those, what shape are they? Did you say pleomorphic? Yeah, many shapes. Really weird bacteria. Really weird. So what is penicillin's target, specific target? Cell what? Now wait, don't answer. Wait till you read all the answers first. Cell walls, cell membranes, enzymes that make peptidoglycan, enzymes that make phospholipid bilayers or DNA. Now, aren't you glad you didn't choose your answer yet? Yeah, it's the enzyme that makes peptidoglycan. That's what penicillin literally binds to. And then that's going to stop cell wall production. Right? She's tricky, that Miss Eden, isn't she? Okay. Why doesn't penicillin work for walking pneumonia? So your patient has been taking penicillin for a month. Nothing's working. Why? How could you explain this? Educate your patient. Because right now they're probably really mad at the doctor. But the doctor didn't know it was Mycoplasma pneumonia. So, okay, so what would you tell them? A, penicillin doesn't target bacterial structures. B, penicillin doesn't work on gram-positive bacteria. C, penicillin doesn't work on gram-negative bacteria. D, penicillin doesn't work on bacteria that lack a cell wall. E, all of the above. You chose D, didn't you? Good. Penicillin isn't going to work on bacteria that don't have a cell wall because there's no PBP target. And they're used to not having a cell wall. They've got something else like sterols. See how clever you're becoming. Yes, you can educate your patients. Yay! Okay. Let's do some other quiz stuff, okay? Based on this gram stain over here, this is a streptobacillus. Would you say this is gram positive or negative? Well, even though a few places look a little decolorized, you're thinking purple, aren't you? Yes, gram positive. So it's A, gram positive. How about this one? It's B, gram negative, right? If it's not purple, it's not positive, it's gram negative, right? Based on cell wall characteristics and envelope characteristics, which bacterial organism has the most, which bacterial organism has more peptidoglycan in the cell wall? Gram positive, staying purple, because they've got more outer peptidoglycan. So that's got to be positive. That is A, gram positive. I can talk. Based on cell wall characteristics, which bacterial organism would be harder to kill with a disinfectant and why? See, now we're starting to discuss why it matters, okay? The why. Did you say gram negative? You're right. And why is that? Because gram negatives have that extra protective plasma membrane barrier. It's semi-permeable. So disinfectants have to get through two membranes in order to hurt the cell, or at least one. Yeah. Based on cell wall characteristics, which bacterial organism would contain an endotoxin that potentially produces fever and shock? Oh dear. Okay, what one is that? Okay, oh yeah, that's the one with the freakish outer membrane. That would be gram negative, the freakish one? Correct. Gram negative, the freakish one. Good. Based on cell wall characteristics and cell envelope characteristics, which organism has the freakish double membrane, unlike any other cell known on this planet? It is B, gram negatives. Very good. Okay. Based on bacterial morphology, which type of bacterium would typically be harder to kill with disinfectants and antibiotics? Okay, so A, gram-positives like Staphylococcus and Streptococcus. Gram-negatives like E. coli and Salmonella, and Pseudomonas aeruginosa, Helicobacter pylori, Treponema, Borrelia, that sort of thing. Or C, acid-fast like Mycobacterium tuberculosis. Now, we haven't done these head-to-head yet, have we? So this isn't nice. She's being mean. Okay, so did you throw out gram-positives? Yeah, their outer peptidoglycan cell walls just sticking out there. So on a surface, staph and strep are actually much easier to kill. Now it's between gram-negatives and acid-fast. Now, the gram-negatives have an outer phospholipid membrane with all that extra layer, but acid-fast have that mycolic acid. So what do you think? You know what? It's the acid-fast. Amongst those three, the acid-fast are the toughest. Harder to kill with disinfectants, harder to strike with an antibiotic. Doesn't mean that staph and strep aren't deadly. They certainly are. In a human, oh yeah. But, because they're very clever in other ways, but on a surface and pound for pound, yeah. Okay. Let's add one more into the mix. Ooh, what about endospores from like Clostridium difficile or Bacillus anthracis? Did you say D, endospores? Right, because nothing is harder to kill on this planet than endospores. So again, think of your patient in the room with C. diff. Okay? Don't freak out. If you have normal microbiota and a good immune system, it's okay. You can fend this off. It only attacks generally people with something going on with their immune system or who have lost their normal microbiota. Okay? But wash your hands. Okay. How about a little bit more? How about DNA? Now, all cells have to have DNA, but prokaryotes, bacteria, like I said, only have one circular chromosome. And here we are here in that nucleoid area. I'm not going to go through DNA replication and all of that. I'm going to kind of cut this one short as much as possible, but there are antibiotic targets that we must talk about here. We're not only going to talk about DNA replication, but then later on protein production. So DNA again, bacteria have only one single circular chromosome. We saw that. The arrow shouldn't be here. It should be on the blue. The chromosome is in the nucleoid area here, but the extra pieces of DNA are called the plasmids. That's where the arrow is pointing. So there's a red and kind of a yellow-orange plasmid. Like I said, plasmids send genes from one cell to another. Remember that? We talked about that happening with the sex pilus, but they can do it even without a sex pilus, but we won't go there. DNA has to occur by replication in prokaryotes, just like with eukaryotes. So DNA replication, they pretty much have the same types of enzymes that do the job, but there are some subtle enzymatic differences. So some of their enzymes are a little different than ours. So their enzymes are different than your enzymes. Would those enzymes in DNA replication make a good target? Yeah, they would. So these different bacterial enzymes are targets for antibiotics. Okay, let's talk about one. Heard of Cipro? Levaquin? Maybe not. If you haven't, these are major antibiotics that are used commonly in hospitals. Okay. So these are drugs that target DNA replication. They're going to target a very special enzyme that, yeah, our cells have them and bacterial cells have them, but bacterial cells have a different enzyme that this drug targets. Both of these drugs are in a class of drugs known as fluoroquinolones. I know that's a tough name. But okay, so these two drugs, levofloxacin or Levaquin. If you know Levaquin, I'm fine. Ciprofloxacin or Cipro. So no, Levaquin and Cipro. Okay? So what are they going to do? These drugs are going to attach to a bacterial enzyme known as gyrase. Now again, human cells have gyrase, but the bacterial gyrase is a little different. So, why is gyrase, so what's it supposed to do? Why is it so important to cells? Gyrase in cells are supposed to keep the replicating DNA molecule from breaking as it uncoils. So here's a bacterial cell, and again, it's supercoiled. And when bacterial cells uncoil, they would snap. But there's this little enzyme here shown in red, and this is our gyrase enzyme. And it's supposed to reattach the DNA after it's been broken, so it just it keeps it together. Now, what happens if a drug attacks this particular gyrase? Well, then the DNA, the replicating DNA molecule breaks. And that's pretty much cell death, okay? So again, gyrase is used in both DNA replication, but also in transcription. When a gene is about to be transcribed and copied in a messenger RNA, DNA has to uncoil there, and it uses gyrase. So understand that drugs that target gyrase are very powerful, okay? So they're very powerful drugs. So the drug basically causes breaks in DNA, and it will kill the bacterial cell. Cell death. All right. By the way, like I said, human cells have a slightly different kind of gyrase. But is it really that different? We have problems with these drugs. Okay? They're not the best targets. Okay? Because bacteria have them and we have them. They're a little different, but maybe they're not different enough. Okay. Ribosomes. All cells have to have ribosomes. Why? To make proteins. But prokaryotes have a uniquely small and different ribosome. Ooh, would that make a target? Sure would. Okay. So we have to look back at ribosomes. Do you remember learning about ribosomes? Okay, think back. DNA has genes. Genes get copied into messenger RNA. That's transcription. And then the messenger RNA goes and finds a ribosome. And then the messenger RNA is translated at the ribosome into a strand of amino acids to make a protein ultimately. So ribosomes make proteins. Okay. So again, the ribosomes, so what do? They assemble the amino acids, and the amino acids are brought in by transfer RNAs, like this green little circle here. And the amino acid methionine, MET here, is being brought in by transfer RNA, and they're being carried into the two parts, to the ribosome that's going to form. So ribosomes are made of two subunits. Now, the bacteria ribosomes are called 70S ribosomes. The S stands for Svedberg. You don't need to know, and it's not going to make much sense to you. But so because the ribosomes are smaller, if they measured it using the Svedberg method, which is a centrifugation, weird thing, don't worry about it. But the subunits, okay, you have a 30S and a 50S. The 30S is the small subunit. The 50S is the large subunit. And if you're trying to make 50 and 30 add up to 70, it won't. Again, it's just an old way that they used to measure molecular sizes. But, okay. But these are different because humans have a bigger ribosome. So again, this is just, oh, I'm sorry. Hold on. Okay. So there's a 30S subunit and a 50S subunit to make a 70S ribosome in prokaryotic bacteria. So the bacteria, like your pathogens, have ribosomes like this, which is a little different than ours. So are there drugs that block 70S ribosomes and mostly leave our ribosomes alone? So there are drugs that block the ribosome, or what goes on in translation here at the ribosome. These drugs, we tend to say, okay, stop protein synthesis. So they stop the process by stopping translation, and usually somewhere around the ribosome. So these drugs usually bind to either the 50 or the 30S portion of the ribosome. One drug, azithromycin. What? Okay, Z-Pak, you guys. Oh, Z-Pak. Hopefully most of you have heard of Z-Pak. Now that one binds to the 50S, portion of the ribosome, blocking this. So what does that mean? No proteins. Okay, no translation of genes. Okay, what about another one? Tetracyclines. Okay, like minocycline and doxycycline. They don't exactly bind to the ribosome, but they stop transfer RNA from getting there. So same deal, no translation if the amino acids can't be brought in by transfer RNA. Consequences: no proteins for the bacterial cell. By the way, if your patient is taking azithromycin or tetracycline and they're non-compliant, meaning they don't keep the concentrations up, that's a problem because this actually doesn't kill the bacterial cell. These two drugs, they just stop them from making proteins and slow them down. If you leave the bacteria there long enough, they'll just make more ribosomes and then start to make more proteins, and then they're fine. So you got to keep the concentrations up until the immune system kicks in. More about that later. How about a question? Oh, we're back to those little Rocky Mountain spotted fever bacteria known as rickettsia, and they're hiding inside of our neutrophil. This is us in blue. How can we poison? Okay, how can we send poison to hurt the prokaryotic bacterial cell without hurting the eukaryotic human cell? Again, these are poisons. These antibiotics are poisons. Answer, again, target unique structures only found in prokaryotic bacteria. Just only their cells have them. What would those be again? Well, PBPs that make peptidoglycan cell walls, the special bacterial gyrase enzyme, and the special 70S ribosome, right? Which would be the safest antibiotic, do you think? All right, so is there some structure in bacteria that our cells do not have, never will? And if you knock out that bacterial enzyme, our cells are like, whatever. Okay, it would be the enzyme PBP, because PBP makes peptidoglycan. Do our cells have peptidoglycan cell walls? So our cells don't need PBPs. Penicillin isn't going to make the slightest difference. Okay? That is the safest antibiotic, assuming you didn't mount an allergic reaction to it. Okay, that's another topic. Let's talk about bacterial toxins. Some bacteria manufacture toxins. So they manufacture them here, sometimes at the ribosome, somewhere else. But then they are released sometimes, not always, okay? There are two kinds of bacterial toxins. Exotoxins that exit the cell, so they're manufactured by the bacterial cell and then released. Exotoxins are mainly released from gram-positive bacteria. Examples, Clostridium, okay? Staphylococcus and Streptococcus. Anybody interested in these two? Anybody have these two and then might be interested in exotoxins that are made? Yeah. Okay, so these toxins that are exotoxins are proteins. So they're manufactured and secreted outside the cell. Exotoxins can be stopped with host antibodies, or if there's no time for the host to make their own immune response and make antibodies, you can give a patient preformed antibodies. Okay? Oops. So here we are. Here's a host cell. Here are toxins that are trying really hard to attach and kill this poor host cell. But if there are antibodies waiting, these little green Y-shaped proteins that we make to protect ourselves, these will actually attach and neutralize any kind of toxins, okay? Now, the second kind of toxin is known as an endotoxin. That tends to mean inside. These are only in gram negatives. So exotoxins, mostly gram positives, but not always. Endotoxins can only be found in gram negative bacteria. Remember LPS? Yes, part lipid, part sugar. We said the lipid part is an endotoxin. It is found in that outer freakish membrane, only in gram negatives. So, to summarize, exotoxins being secreted out, endotoxins. Now, if this is inside the cell, located in that outer envelope just outside the cell wall in that freakish membrane, when are these guys released? Well, if the bacterial cell starts to die and break up, it releases more endotoxin. So when we give sometimes patients antibiotics that kill gram negatives, they will start releasing more endotoxin and the patient will get more symptoms. Yeah. Weird. Okay. I know, graphic here. So let's talk about exotoxins for a moment. Exotoxins, like I mentioned, are proteins and they are very specific. They attach to different cells and have different damaging effects. Because they're being released out of the cell, they can be located far away from the original bacterial cell that made it. So the bacteria don't have to be present to cause disease. So here we are. It could be the toxins can be secreted out into the food. They are secreted. A lot of times they cause something called foodborne intoxication. If you ingest them, then there you go. And the thing is, you can take this home and heat it up, but if the toxins are already there, some of these are very heat stable. Okay? So again, exotoxins tend to be very specific. They attach to specific cells in the human. So some of these exotoxins are neurotoxins, meaning they attach to nerve cells and damage the nerve cells and therefore the nervous system. Can cause paralysis. Some of these exotoxins are all attached to the intestinal cells. We call those enterotoxins. They attach to intestinal cells and cause intestinal disturbances. This is what we call the double bucket flu. It's not really flu. Flu is a virus, but there you go. Cytotoxins, okay, well, this means that these toxins attach to and cause damage to a variety of cell types. Many toxins, by the way, are fatal before the immune system has a response and makes protective antibodies. So sometimes it's too late, which is why we try to have preformed antibodies to save them. Okay, this is busy, I'm sorry, but this is all FYI. Some of you have bacteria that make exotoxins, and your exotoxin, when you're reading about those, you might read, oh, it's an AB toxin. What does that mean exactly? So I'm going to describe how this happens in Corynebacterium diphtheria. This is a bacterium that causes diphtheria. So this won't be on the test, but you might want it for your paper. So here's the bacterial organism that manufactured the AB toxin. There's an A subunit and a B subunit. It is released because it's an exotoxin. And it finds the specific receptor on the specific cell that it is supposed to affect. So again, they're very specific. Ones that attack nerve cells don't attach to gut cells, the ones in the intestines, okay? But this one attaches to a whole bunch of cells. So it attaches and it's brought in by something called receptor-mediated endocytosis. Breaks apart. The A is active. Well, I should let you know the B stands for it binds. It binds to the cell receptor, and then it breaks apart, and the A part is the active part. And so again, it is the active part, the active enzyme that's going to hurt the human cell. So this is your cell. It got into your cell. How is it going to hurt your cell? Turns out this little subunit here of the toxin attaches to your ribosome and stops protein synthesis. You know how we make drugs to kill bacteria by blocking their protein synthesis? Well, they make toxins to kill our cells by blocking protein synthesis. Whoops, that one's supposed to, okay, whoops. I thought I had another image there, but there you go. All right. There are other exotoxins. Some, by the way, damage membranes, and these are membrane-damaging toxins, like hemolysins. When we talk about group A strep, which is caused by the species Streptococcus pyogenes, they produce an enzyme called a beta-hemolysin enzyme, beta-hemolysin, that when it gets to a red blood cell, it causes that red blood cell to blow up. So these are sheep red blood cells in agar, and they grew up Streptococcus pyogenes. They released their toxin, and it caused the red blood cells to explode. Some bacteria, some other members of the strep group will only cause the red blood cells to leak out. But here we have Streptococcus pyogenes attached to a red blood cell. So it would release the toxin and cause it to lyse, break apart. Now, if this was inside your body and all these toxins were causing your red blood cells to break apart, that would be bad. And that's only one toxin that group A strep or Streptococcus pyogenes can make. Some exotoxins are super antigens. Okay, they're super in that they're going to really make you sick. So we call them super. What is a super antigen exactly? Here's a Superman for super antigen. It is any antigen that activates a huge portion of the immune military force. Now, if you're looking at this image here, this is really complex, but this is something like these are two white blood cells, and they have to shake hands and talk to one another. Usually when this guy encounters a normal antigen, it presents it to this white blood cell, and they shake hands and they talk to one another using chemical signals. And a regular antigen causes a normal conversation and says, you know, we need to have a battle here. There's an invader, so we're having an immune response. That's what antigens do. Antigens generate an immune response. So this gets these two white blood cells kind of going, and they get other white blood cells going, and there you go. Now, a super antigen can stimulate 5 to 30% of your major what we call the white blood cells, okay, the T white blood cells. Normally, a normal antigen only affects maybe 0.001% of your military force, but a super antigen stimulates 5 to 30%. What does that mean? So there's your super antigen. Okay. So you're going to get a huge signal, like we call this a huge immune signal storm, or even more technically, a cytokine storm. The conversations going on between these white blood cell soldiers just basically tells the body that we're under attack and we need to set off nuclear weapons. That's how bad this is. So when there's a super antigen, the body's immune system goes so crazy. You get fever, shock, and sometimes a rash, but it can result in death. This is what toxic shock syndrome is and necrotizing fasciitis. So this is what's going on. This is because the immune system is freaking out with 5 to 30% of the white blood cells going. Who does this of the bacteria? Staphylococcus aureus and Streptococcus pyogenes. Group A strep. Okay. Toxic shock syndrome. This was really bad when women were wearing, they were using high absorbent tampons. Sometimes the bacteria are in the vaginal area, and a tampon unfortunately traps a lot of blood and nutrients and bacteria. So women were getting all of these super antigens, and some died. Some other toxins, exotoxins. Clostridium botulinum, for example, releases an exotoxin that causes flaccid paralysis. Now, the exotoxin is a neurotoxin. That means it attaches to a nerve cell, causing paralysis. So here's what we know. So the bacterial exotoxin, known as the botulism toxin, is released from the bacterial cell. And what is it attached to? It attaches to a neuron, one of the motor neurons, that's supposed to tell your muscle cells, specifically those peripheral muscle cells, to contract. Okay, so it attaches to the neuron, the motor neuron, stops the motor neuron, the cell, from releasing its vesicles that contain neurotransmitters. Neurotransmitters, I can say it. The neurotransmitter in question is acetylcholine. If neurotransmitters aren't released, okay, this doesn't signal the muscles to move. So again, you have paralysis. Remember your anatomy. So no acetylcholine means no signal to the muscle cell, means paralysis. And it's a peripheral paralysis. So you get, unfortunately, floppy baby syndrome, and that is really a name. It's awful. But you get, for people adults who get it, they have droopy eyelids. Maybe they just are very, very droopy. But this can also cause the peripheral nerves of your diaphragm to stop working, and that means you're not breathing, right? We like our diaphragm. Clostridium tetani also, its cousin, releases an exotoxin. This causes rigidity and spasms. The reason for that is, now it still binds to the peripheral nerves, but then it moves to the central nervous system. Okay, it also blocks a neurotransmitter, but in this case, it's gamma-aminobutyric acid, or GABA. Remember that from anatomy and physiology. GABA release is needed to normally inhibit your motor neurons. So this is sort of the opposite. You get these violent spasms that will literally break bones and cause lockjaw. So this is why we need vaccines, because, by the way, we're not going to be able to form antibodies against the toxin in time. So you need to have an immune response ready to go, which is why we have our tetanus shots. How about E. coli? You know what? They are gram-negative, but they make both exo and endotoxins. Okay? So they can only do this, though, if you're thinking, but I feel okay. Well, E. coli can only make an exotoxin if it got the gene instructions from maybe a friend. So remember, E. coli always has LPS. So that's an endotoxin here, and when they break apart, they can release the endotoxin. Now, some E. coli might have genes to make some nastier LPS endotoxin. That is true. Now, it turns out, and by the way, LPS is very heat stable. So if you're thinking you'll just heat your food up and kill E. coli, no. You better actually, you need to just turn it into charcoal. Now, there's also exotoxins being made by E. coli. These can be heat stable and heat unstable. Okay. Now, where did E. coli get the really nasty genes to make an exotoxin? Well, probably from its environment. It got a hold of a toxin gene originally found in a bacterium known as Shigella. Shigella is like Salmonella, only usually worse. Okay. The consequences of getting that, if the E. coli has the Shigella gene, it makes the Shiga toxin. Shiga toxin. And, okay, it can be released by E. coli. Now, there are some other gram-negative bacteria that also will grab this toxin. What does this toxin do? The problem is that because it's an exotoxin, it can leave where E. coli is hiding out, and it can end up in the kidneys. Now, the bacterial cell, E. coli, or any other, the bacteria are not in the kidneys, but there's going to be kidney damage. Why? Because the toxin is in the kidneys. What does the toxin do? It attacks the protein synthesis in the kidneys, and you get dead cells in the kidneys. and you get something called hemolytic uremic syndrome, which is bad, which will put you in dialysis, or your patient. When a patient gets E. coli, especially if they start urinating blood, this is very serious. And when the doctors tell that patient to drink water, it isn't just because of the diarrhea. You have got to flush the kidneys out. You've got to get the toxins out of there. All right? Okay. You need to flush the toxins out. This is the solution to pollution is dilution. Because your patient, I'm guessing, does not want to go on kidney dialysis. All right? So this is the Shiga toxin released by E. coli. So it's an exotoxin. It's released by the bacterial cell. It attaches to the kidney cell. It is brought in. It is processed by the kidney cell factories and sent over to the ribosome where it stops protein synthesis. And there you go. So it's not, we want you want your patients to drink water, and it's not all about dehydration. It's about flushing out the toxins. Again, it's not the bacteria here, it is the exotoxins. Have I repeated that enough? Good, it could save a life. Okay, we're going to finish up by talking about eukaryotes and quickly viruses, really quickly, really quickly. Okay, the morphological characteristics of eukaryotic cells. Here are our neutrophils again, our white blood cells. Why are they different than the bacterial cells? Okay, pretend for a moment that this cartoon is actually one of your white blood cells in your body, one of your immune soldiers. It's eukaryotic. All of your white blood cells are. Okay? So what does it have? It's eukaryotic. It has a true membrane-enclosed nucleus with multiple linear chromosomes. Don't worry about number 1. Okay, so DNA replication is by a different gyrase enzyme than with bacteria. And look at all these membrane-enclosed organelles that we've got, 3 through 12 pretty much, okay? 5 is the rough endoplasmic reticulum. It's called, it's basically a factory, and it's called rough because it has little ribosomes attached to it. The ribosomes are 80S. What are the bacterial ribosomes? 70S. So, what do you think these factories make with the ribosomes attached right to them? Proteins. So the rough endoplasmic reticulum is involved in manufacturing protein products after translation, right, at the ribosome. Then you have a smooth endoplasmic reticulum here. Its job mostly is to manufacture lipids, but to do a whole bunch more for the cell. We won't get into that. You have the powerhouse of the cell, the mitochondria, or mitochondrion is singular. So the powerhouse of the cell. And we feed it food and oxygen, and we do that if we want to have enough ATP energy. So if you want your white blood cells to have energy, there you go. What if your patient is anemic or not getting enough oxygen? That means your white blood cells aren't going to be doing their job, right? You need to feed your mitochondria food and oxygen to make power for the cell, for your white blood cells to go and fight and do their jobs. One thing I'm going to mention a little bit later on, mitochondria have a double membrane, 70S ribosomes, and a lot of characteristics that are prokaryotic. It is a long topic I can't get into, but the theory goes, it's called the endosymbiotic theory, that before eukaryotic cells became complete, they took in bacterial prokaryotic-like cells. Okay, this will come up later. All right, and then we also have things called lysosomes. Lysosomes contain powerful digesting enzymes called lysozyme. Yay! So this can digest things that need to be digested or broken down in the cell. Yay! Breaks up bacteria too, because, well, this is a white blood cell. and they tend to many times munch up bacteria. And then the lysozyme in the lysosome will break them up. Want a picture of it? Okay, here's a white blood cell, a phagocyte, bringing in fluorescently green E. coli. So it's being brought in by phagocytosis, and this white blood cell, our white blood cell soldier, releases lysozyme to break down E. coli. Hey, why don't these bacteria die? The rickettsia, in fact, is living inside our white blood cells. Why aren't they being digested? Turns out some bacteria want to be taken into the cell. Being taken in or phagocytized does not mean digested. Some bacteria actually block those lysosomes and are not digested. They even thrive inside the cells. They replicate, and many times they need to live inside of our cells. We call these kinds of pathogens obligate, intracellular pathogens. Break it down. Obligate means they have to. Intracellular means in the cell. They have to be in the cell. Examples. Rickettsia rickettsii that causes Rocky Mountain spotted fever. And also Mycobacterium tuberculosis. They actually love to be eaten by something called a macrophage. It's a white blood cell. It eats it, but is unable to digest it with lysozyme. And then the bacteria, the tuberculosis bacteria, move around the body inside the white blood cell, never being detected. And it causes tuberculosis, right? Right. Lastly, viruses. I know I cut out the majority of this material, but we still need to cover a couple things, and then I'm also going to do a quick review. So, viruses are not living at all. Some argue, though, that they become living once they take over their host cell. And they kind of are, you guys. They're like robots. Think of viruses like robots. This is a herpes virus, just as an example. We call the infectious particle of the virus that goes out of one cell into the next, it's called the virion. It is the actual infectious particle. What makes up a virion? Glad you asked. Okay, so first of all, you have a protein coat in green. It's known as a capsid. You have some kind of nucleic acid, either DNA or RNA, here in red. Okay, and by the way, if you put the nucleic acid plus the capsid together, it's called the nucleocapsid. So both the red and the green. Okay. Some viruses have an envelope made of a phospholipid bilayer. Here it is in gray. Some viruses have little spikes in orange. This allows them to attach to host cells. It's like landing gear. We call these little spikes papillomeres. Some viruses have unique enzymes that help them in their invasion process. Again, viruses, at least the virion part, does not metabolize, move, or grow. They are assembled inside of a host cell, inside of you. Once they get in, they take over. They take over your factories using their DNA, and your cells end up making more viruses. These viruses tend to be very specific about which type of host cell they infect. Again, they don't move, they just bump into a host and grab on by receptors. Here is one of your cells, and here is a herpes virus. So again, once it enters the cell, it becomes your cell. Your cell may have some cell functions going on, but its primary function tends to be making more viruses. So how do you kill a virus without killing the host cell? It's not easy. Well, can we target unique enzymes that might be a target? I said that some viruses carry unique enzymes. Yeah, if you can find the enzymes, but that takes research. Some drugs are going to mutate the DNA replication process in the cell. Yeah, it's going to hurt the host cell, but it should hurt the replicating viruses more because they're making DNA faster. That doesn't sound good. We'll talk about that later on. Now, the rest of this is review, okay? So I know I'm running out of time. So if you want to pause and come back, remember that you do not have to read a textbook. It's all in the lecture. So the lectures tend to be a little more lengthy. But as a treat, I follow everything up with little quizzes, okay, to help you learn the material. So it's up to you whether or not you listen, and I hope you do, to the entire lecture and go through the quizzes with me. And if you listen, I will tell you the answers, and I will tell you why those answers are right. And I will even tell you why some of the answers are wrong. Doing that, you're going to do better on the test. And also writing your outline and research paper. Okay? In fact, most of you have a lot of material that you can put into your research topic. Are you ready? Okay. So, first question, what do you call extra round pieces of bacterial DNA that are not chromosomal? Pili, fimbriae, plasmids, mesosomes, gyres, transposons. Some of these things you've never heard of before. You think it starts with a P, but which one is it? No, pili, that's like a sex pilla. So plasmids would be the right answer. Oh, sorry, plasmids. So C is the right answer. How do bacteria keep from being phagocytized or taken in by host white blood cells? By A, pili, B, capsules, C, plasmids, D, porins, or E, they taste bad to the white blood cells. And again, this is a big virulence factor. Capsules, the glycocalyx. It makes them slippery and invisible to the immune system, to white blood cells. So that would be B, capsules. How do gram-negative bacteria have sex? So here's a couple of bacterial cells, and they're sending over sex. Pili, fimbriae, flagella, pseudopod, cilia. Pili, a sex pilus. That would be A. Very good. So what would be the approximate size of a bacterial cell? 3 centimeters, 3 millimeters, 3 micrometers, 3 nanometers, 3 yards. No, this is medical microbiology. It's got to be a micrometer. It would be number 3, 3 micrometers. Okay. What makes peptidoglycan cell walls? The enzyme known as gyrase, the enzyme known as lysozyme, the enzyme known as PBP, the enzyme known as penicillin, many types of enzymes, or ribosomes, which are like enzymes actually. Peptidoglycan, PBPs, peptidoglycan building proteins. It's C, right? Okay, not gyrase, that's involved in DNA replication. Lysozyme is the enzyme that digests stuff inside of our cells. So you, like white blood cells have this. And penicillin is what blocks the PBP, all right? And not ribosomes. Okay, ribosomes make proteins only. LPS is a, or an, well, I guess it'd be an exotoxin. Oh, hold on. Endotoxin, exotoxin, and endotoxin. Exotoxin and antigenic structure on gram-positives. Endotoxin and antigenic structure on gram-negatives. Which one has the freakish outer membrane? An LPS. That would be gram-negs. And again, it's an endotoxin. And part of it's the endotoxin and part of it is sticking up, and so it's antigenic. It generates an immune response. Okay? What is this bacterial shape and cell arrangement? Diplococcus, Diplobacillus, Streptococcus, Streptobacillus, Staphylococcus, Staphylobacillus, Streptospirillum, Staphylospirillum. Here's one cell. Here's another. Here's another. Well, it's a bacillus, and it's in a strepto formation, so BD, streptobacillus. Okay. What process does azithromycin Z-Pak inhibit? Oh, DNA synthesis, peptidoglycan cell wall synthesis, peptidoglycan cell membrane synthesis, protein synthesis. It is D, protein synthesis. Okay, it attacks the 50S portion of the ribosome, stopping translation of messenger RNA at the ribosome, stopping the bacterial cell stopping the bacterial cell from making proteins. Okay, that's what Z-Pak does to the bacterial cell. What process does penicillin inhibit? DNA synthesis, peptidoglycan cell wall synthesis, peptidoglycan cell membrane synthesis, protein synthesis. Penicillin gram-positive, stain purple because they've got peptidoglycan. Yeah, but is that in the cell wall or cell membrane? Which one? B, cell wall. Okay, so penicillin inhibits peptidoglycan cell wall synthesis by attaching to the PBP. The target is the PBP enzyme. What process does ciprofloxacin and levofloxacin, what process do they inhibit? DNA synthesis, peptidoglycan cell wall synthesis, peptidoglycan cell membrane synthesis, protein synthesis. Yeah, that was the DNA synthesis one, right? Yeah, okay. Remember, gyrase. Okay. Which toxin is a protein that could be stopped by antibodies if only the host could make them in time? Exotoxin, endotoxin, or both? A, exotoxin. Yeah, it's released. Okay. Which type of toxin is LPS? A, exotoxin, endotoxin, or both? LPS. It is an endotoxin in the freakish outer membrane of gram-negatives. Right. LPS is associated with gram-positives, gram-negatives, both neither. Yeah, I know. I already told you. B, gram-negative bacteria. Which antibiotic will work on this? This is a virus, Miss Eden. Penicillin, tetracycline, Z-Pak, Cipro, Levaquin? None of the above? Yeah, you said none of the above. But do you know why? Penicillin only attacks enzymes that make bacterial peptidoglycan. This doesn't have a peptidoglycan cell wall. Tetracyclines and Z-Pak stop the ribosome. Well, basically, this stops the transfer RNA. This one stops the 50S portion of the ribosome. So both of these stop protein synthesis. Viruses don't make proteins. Cipro, Levaquin. Well, they stop DNA replication by using the bacterial gyrase. These things don't replicate their own DNA. They rely on the host, right? Host does everything. This is like a robot. It can't manufacture itself. It has to take over a cell, use the cell factories. It uses its own DNA to do that. So when your patient demands an antibiotic because they have a cold, yeah, won't work. There's nothing for these drugs to attack here. And if they did, they would attack your cells or their cells. So that would be bad. All right. Two men walk into a bar. The first orders some H2O. The second one says, sounds good, I'll have some H2O too. Second man dies. You have to think about that. What is H2O2? Well, that's hydrogen peroxide, you guys. Anyway, Cat's done. My cat is alive. I was just joking. So email me if you have any questions. Good luck on the assignment. I've pasted it here, but I will also, of course, put it online. So, and here's the rubric. You guys have a great week. Let me know if you have any questions. This should be great for helping you along with your project, okay? It's a lot, I know, but you didn't have to read, okay? You didn't have any book reading assignment. So, and I'll try to put a practice quiz up on online again for you too, okay? All right. Take care.